Hello everybody. Today we are going to discuss Unit 14 of Organic Chemistry. The chapter is Biomolecules. Let us get ourselves introduced to biomolecules. Now dear students, before we go about the chapter, I want all of you to be ready with your notebooks and pen so that you could take down certain points when we go through the presentation. So, are we ready? Then let's get started. It is known that all living systems need matter as well as energy for their growth and maintenance. This is a very remarkable character of any living system. The ability to grow, to sustain, and to reproduce itself requires the most important component, energy. And where does this energy come from? It comes from the food what organism would eat. So the food and hence the components and constituents of food is very important for a living organism. So a living cell consists of about 50 different elements, out of which 11 elements are most abundant in the living cell. Which are these different elements? They are carbon, hydrogen, oxygen, nitrogen, calcium, phosphorus, potassium, sulfur, chlorine, sodium, and magnesium. Isn't it amazing, students, that a living cell is made up of such non-living atoms and molecules? Indeed, it is the wonder of nature. Out of all these 11 most abundant elements, carbon, hydrogen, oxygen, and nitrogen account for about 98% of the total mass present in man. 70% of the weight amounts to water. Then, in addition to water, there are large number of carbon compounds. It is these carbon compounds which we are all focusing our study in this particular chapter. Whenever we are talking about these different carbon compounds, then we have to consider the various chemical changes which take place in a living system. These chemical changes associated with these compounds fall in the domain of biochemistry. Biochemistry involves the study of cells at molecular level. Biomolecules are a group of complex chemical substances that naturally occur in all living organisms. They are namely carbohydrates, proteins, lipids, hormones, enzymes, vitamins, nucleic acids, and the list continues. Based on their molecular mass, the biomolecules are divided into two classes. What are they? They are divided into micromolecules and macromolecules. Micromolecules have a molecular mass in the range between 100 to 1000. Some of the examples listed under them are sugars, lipids, vitamins, etc. Macromolecules are the compounds with molecular weight greater than 1000. The examples include proteins, polysaccharides, nucleic acids, etc. So, all these biomolecules constitute the molecular logic of life. Let us now focus on the most abundant biomolecule on planet Earth, carbohydrates. Historically, carbohydrates were defined depending upon the ratio of hydrogen and oxygen atoms that was found in their molecules as 2:1, which is very similar to the ratio found in water. So, these class of compounds which had hydrogen and oxygen as a ratio of 2:1 and also contained carbon were called carbohydrates because carbonate dehydrate is the Latin word used for the meaning hydrate of carbon. Hence, that is how the name was coined for carbohydrates as hydrates of carbon. So, they were given the general formula CnH2ON. But there were some exceptions. Let us take them one by one. Acetic acid is one major exception. Acetic acid has formula CH3COOH containing 2 carbon atoms, 4 hydrogen atoms and 2 oxygen atoms. So, the ratio of hydrogen and oxygen no doubt is 2:1, which is similar to what is found in water. But I'm sure all of us agree that acetic acid is not a carbohydrate. Acetic acid is belonging to the class of organic compounds called carboxylic acids. Similarly, let's talk about the other case study done with rhamnose. Rhamnose is a carbohydrate. It has formula C6H12O5, but it does not have the ratio of hydrogen and oxygen as 2:1. Thus, even though it is a carbohydrate, it does not fit into the definition. This was very confusing to people who were studying carbohydrates, and I'm sure you also would be wondering what is then the definition that could be given to carbohydrates. Finally, it was settled down as the modern definition of carbohydrate to be completely framed based on the functional groups that are present in carbohydrates. On analysis, it was identified that carbohydrates contain two important functional groups. They could have aldehydes and alcoholic groups, or they could have ketones and alcoholic groups. So therefore, the definition was devised as the optically active compounds, which are polyhydroxyaldehydes or polyhydroxyketones, are called as carbohydrates, or the compounds which will produce these units on hydrolysis are also called as carbohydrates. Let us analyze the definition. Now, optically active compounds. These are the compounds which have the ability to rotate the plane of polarized light in either clockwise or anti-clockwise direction. Such compounds are called optically active compounds. Now, we also have a terminology, hydrolysis. Now, what does it mean? Hydrolysis means breaking a substance with the help of water. So, carbohydrates are those compounds which on hydrolysis, that is breaking down with water, could produce polyhydroxyaldehyde molecules or polyhydroxyketone molecules. So that is how the definition was devised for carbohydrates. I hope students are understanding and it's clear to them with regard to the definition. Now, the most important and pertinent question which could be crossing all your minds would be, why should I study carbohydrates? What was the need to understand these class of biomolecules in such greater depths? Now, let me throw some light on this particular question. Carbohydrates are vital source of three basic necessities of life: food, clothing, and shelter. I have also included photosynthesis because it is a very important phenomenon which occurs and is also one of the primary reasons for the formation of carbohydrates. On planet Earth, carbohydrate is found to be the most abundant biomolecule because it is primarily produced by plants during photosynthesis. We are aware about the preparation of glucose by plants using carbon dioxide and water and sunlight, and the phenomenon is called photosynthesis. So this is a remarkable, very important reaction going on planet Earth, producing this important biomolecule, carbohydrate. What is very interesting is every year plants convert more than 100 billion metric tons of carbon dioxide and water into cellulose and starch. Both cellulose and starch are found in plants. Indeed, so much of carbon dioxide is converted into useful product called carbohydrate by all these living or lungs of planet Earth, the green plants. In addition to this, carbohydrates in the form of insoluble polymers, they serve as structural and protective elements in the cell walls of plants and bacteria. They are also found in the connective tissues of animals. Nucleic acids, the very essence of life, contains ribose and deoxyribose as the sugar component in it. So indeed, we need to understand carbohydrates very well because the very logic of life is completely revolving around carbohydrates. So let us understand the carbohydrates better through their classification. So let us get started. Carbohydrates are broadly classified into two classes based on their taste. And the word saccharide used here also comes from the Greek word saccharum. Saccharum means sweet to taste. So therefore, carbohydrates are classified based on this as sugars and non-sugars. Sugars are sweet to taste. Non-sugars are tasteless. Sugars are crystalline compounds. Non-sugars are amorphous substances. Sugars are soluble in water. Non-sugars are insoluble in water or sparingly soluble in water. Non-sugars are also called as polysaccharides. Now, coming to sugars. Sugars are further classified into two different types: monosaccharides and oligosaccharides. Now, what do you understand by monosaccharides? They are the simplest carbohydrates and they cannot be further broken down. Monosaccharides are further subclassified into aldoses and ketoses depending on the function group present in the molecule. If the compound has aldehyde, they are called aldoses. If the compound has ketone, they are called ketoses. Students, I would like to draw your attention. We just understood the definition of carbohydrate where they contain aldehyde or ketone along with hydroxyl groups in their compounds. Oligosaccharides are the type of carbohydrates which on hydrolysis would give few monosaccharide units. The term oligos means few. So the term has been given as oligosaccharides. They are further subclassified as disaccharides, trisaccharides, and tetrasaccharides. Let us understand the classification of these carbohydrates individually in the next slide. Monosaccharides. These are the carbohydrates which cannot be further hydrolyzed to give simple compounds, and they are called as therefore monosaccharides. We are aware of the existence of about 20 monosaccharides in nature. Some of the common examples are glucose, fructose, galactose, ribose, etc. All these come under the class of monosaccharides. Monosaccharides can also be further studied and classified based on the number of carbon atoms that could be present in the molecule. So let us see how are these classified. If you are having 3 carbon atoms and they have a general term given as triose, and if the compound contains aldehyde as the functional group, then it should be called aldotriose. If it has got a ketone as a functional group, it is called then as ketotriose. So students, let us understand once again, if there are 3 carbon atoms, they are called trioses. 4 carbon atoms, tetroses. 5 carbon atoms, pentoses. 6 carbon atoms, hexoses. If there is an aldehyde, then they are all aldoses. If there is a ketone, they are all ketoses. So again, aldotriose, they are having aldehyde functional group and 3 carbon atoms. Example, glyceraldehyde. Ketotriose, they have a ketone and 3 carbon atoms. So example, dihydroxyacetone. Aldotetrose, they have 4 carbon atoms and an aldehyde functional group. Example, erythrose. Ketotetrose, they have 4 carbon atoms and a ketone, so they are erythrulose. Example, if there are 5 carbon atoms and they have an aldehyde, they are called aldopentose. Example, ribose. If they have 5 carbon atoms and a ketone, then they are called ketopentose. For example, ribulose. If they have 6 carbon atoms and an aldehyde group, they are called glucose, example, and called as aldohexose. If they have 6 carbon atoms and a ketonic group, they are called ketohexose, and the example is fructose. Now students, please make note, a very popular question for an example for ketohexose. It is fructose. It is the only monosaccharide which has a ketone and 6 carbon system, and it's studied in our syllabus. So students, this table you need to practice and memorize the names of the respective compounds with the respective examples given in that particular table. Let us proceed towards oligosaccharides. Oligo means few. So, carbohydrates which on hydrolysis will give few monosaccharide units are called oligosaccharides. And how many can they be? They could be somewhere between 2 to 10 monosaccharide units. Hence, the compound is called as oligosaccharides. Oligosaccharides are further classified as the dependent factor being the number of monosaccharide entities obtained from them due to hydrolysis. The first one is called disaccharides. As the name itself suggests, these are the carbohydrates which on hydrolysis would give two monosaccharide units. For example, sucrose C12H22O11. When they are hydrolyzed with water, they produce glucose C6H12O6 and fructose C6H12O6. Maltose. It is also a disaccharide C12H22O11. On hydrolysis with water and an acidic medium, it will produce only glucose molecules C6H12O6. Lactose. It is on hydrolysis yielding a molecule of glucose and a molecule of galactose. Thus, the disaccharides listed, sucrose obtained from cane sugar and lactose present in milk, both are called heterodisaccharides because they produce different monosaccharide units on hydrolysis. Sucrose produces glucose and fructose. Lactose produces glucose and galactose. Whereas maltose obtained from malt is called a homodisaccharide because on hydrolysis it produces only molecules of glucose. So this is how we understand disaccharides. Proceeding towards trisaccharides. These are the carbohydrates which on hydrolysis produces 3 monosaccharide units. Let us take an example. Raffinose C18H32O16 on hydrolysis with water produces glucose, fructose and galactose. So 3 different monosaccharides obtained from raffinose, a trisaccharide. The next is called tetrasaccharides, carbohydrates which on hydrolysis will give 4 monosaccharide units are called tetrasaccharides. For example, stachyose is the example listed under tetrasaccharides. Polysaccharides. Poly means many, saccharum means the taste factor. So polysaccharides are the carbohydrates which gives large number of monosaccharide units on hydrolysis. Now, polysaccharides are belonging to the class called non-sugars. So they are not sweet to taste. They are non-sugars. So we take care to write the definition as carbohydrates which gives large number of monosaccharide units on hydrolysis are called as polysaccharides. For example, starch, which is the storage polysaccharide in plants. Cellulose, the structural polysaccharide found in plants. Another example, glycogen, which is the storage polysaccharide in animals. Now, students, sugars can also be classified into two other separate classes depending on their chemical behavior. They are reducing sugars and non-reducing sugars. Now, what are these? Reducing sugars are those sugars which have the capacity to reduce Tollens reagent or filling solution when they are treated with such reagents. So, what do you understand by these? They are the sugars which can reduce filling solution. The students who have studied biology in first years would have performed this experiment. Filling solution is a blue colored solution of copper sulfate along with sodium potassium tartarate mixed to it and sodium hydroxide. To that, if you will add any reducing sugar and heat it, the blue color changes to red. This indicates reducing property of the sugar. Now, why can they do this? This is possible because reducing sugars have free aldehyde or ketonic functional groups. That means the aldehyde group, if it is present in the compound, it can reduce the filling solution or Tollens reagent, and that aldehyde or ketonic group should be free in the structure of the sugar. Such sugars are called reducing sugars. Students, please note this. All monosaccharides, whether they are aldoses or ketoses, they are all reducing sugars. So you can write examples for reducing sugars as glucose, galactose, fructose, maltose, lactose, etc. The other class is called as non-reducing sugars. Here, the non-reducing sugars do not reduce Tollens reagent or filling solution. The reason they do not have free aldehyde or ketonic functional group in their formulae. Hence, they are all having no ability to react with filling solution or Tollens reagent. They are all called non-reducing sugars. There is a very classical example for non-reducing sugar. It is sucrose. So please keep this example in your mind. It's a very popular question as well. An example for non-reducing sugar is sucrose.